We have programs underway. I think it's great that especially for cell therapy, people have developed these systems where it's all self-contained. You start with the patient sample and you end up with the drug to give back to the patient. And yes, we want our chromatography as part of that for the purification step and used in that process. Continuous chromatography is very much of interest in the world today. We're giving a presentation at the International Conference on Integrated Continuous Bioprocessing coming up in a few months. So yes, one opportunity that is growing. Also, again, the new therapy modes that are growing. There are many opportunities in this industry.
Welcome back to part 2 with Scott Wheelwright, who is the chairman, CTO, and co-founder of BioChromatographix International. If part 1 opened your eyes to next-generation chromatography, this session delivers the practical insights you need. We're exploring how to assess monolithic chromatography media technology for your applications, and Scott's hard-won lessons from building companies across continents and his roadmap for downstream processing innovation. From cell and gene therapy scale-up to actionable implementation strategies, let's dive into what's next for purification technology. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. What were some pushbacks you experienced? So yes, as you said very well, Scott, there are reasons why the biotech industry is conservative. From my experience, when I worked in technology innovation several years ago, well, we had great technology, but then often we had pushback from people working in manufacturing. So I'm curious, what kind of pushbacks did you experience or some questions you got from your early users?
The users, of course, are the final arbiters of your quality. If they don't like it, they won't use it. If it helps them, they'll use it. The issues are, does it work? Is it reliable? Is it consistent? Is it robust? I had many discussions over the years. As a manufacturing specialist, our focus is on making the product and getting it to market as quick as we can at the lowest price possible for the benefit of all the users. Discovery doesn't have the same goals. Part of the mindset is, we're scientists. We want to know how things work. You don't always have to know how it works in order to make it useful. In manufacturing, we want to get it into people's hands, get it used. We can't answer all the questions today. We're looking for improvements. We're looking for feedback from our clients to help us to make improvements. We anticipate several generations of our product. We'll always be improving our product. Our product today is an improvement of what's on the marketplace, and then we expect to continue to make improvements as we learn from our customers what they need and as materials change, as we talked about before. The advancement of technology is mirrored by the advancement of materials. It's just amazing to me how much. So, for example, in the metals industry, how many more formulations there are for metals? 10,000 years ago, there were maybe 5 formulations for bronze, and that was it. And now, how many formulations we have? We have tens of thousands of metal formulations, and we have tens of thousands, maybe hundreds of thousands of formulations. And we can change these properties. Everything's a trade-off. There are limits. Nature sets limits, but we need to understand these limits and how to get as close as we can to those.
Besides getting in front of your users early and having these conversations, what other strategies have you found effective for bridging this gap between the innovation and a commercial product, which can be a very difficult process, a very difficult journey?
Well, I agree. I think that innovation and conversion from a research idea to a commercial product is every bit as difficult as the original innovation. I always liked the holiday parties at the companies I worked at because people would have a little to drink, they'd relax a little bit, they'd be a little more open, a little bit more honest. I remember one researcher coming up to me, he said, "Scott, why are you in manufacturing? You could be in research." I said, "Peter, why would I want to be in research? Research is boring. You do the same experiment 20 times before you get it to work. And we're dealing with millions and hundreds of millions of dollars worth of equipment and it's big scale. And it has to work the first time and it has to keep working for years." And I say, these are real challenges. These are real budgets, timing, all of this. We just keep going until we overcome these. I look forward every day. It's another challenge. It's an opportunity.
What is your vision now as you've recently started this startup company? Where would you ideally like to go? What technologies are you developing? How would you like to expand serving other customers? How will that look like?
Well, our goal is to provide solutions to companies that are facing challenges with their separations and purifications, particularly of the nanoparticles, viruses, exosomes, mRNA, these types of things which have not been adequately addressed by the existing technologies. We have many collaborations with partners all over the world. Our goal is to work with them. For example, we have one collaboration with a company in Europe that is developing AAV, which is the virus that's used in gene therapies. And they have asked for our collaboration to improve their manufacturing process. As shown, we have data that shows that with our materials, we can separate. So one of the problems when making viruses is that when they're made from starting with genetically engineered plasmids, sometimes they assemble correctly with DNA or RNA in the middle, and sometimes they don't assemble correctly, and they're empty. We call these empty capsids, and there's a challenge because there's not much difference. And again, the hardest separations are between two things that are very similar. And an empty virus capsid that has the DNA or RNA inside, depends on the virus which it has, but one that has the DNA inside, this is a difficult separation. And we have successfully separated these. And so we're collaborating with this partner. They make it, we will help them to improve their purification process, and therefore they'll have a lower cost, a lower price manufacturing process, which will hopefully give them an advantage in the market with their product. We have another company that makes plasmids, and we're working with them to improve their plasmid production. We have other collaborations with people who are doing other types of advanced medicinal product, nanoparticles, to help them to solve their problems. We're always looking for a way to, what can we do to improve the quality of their product and to reduce their cost?
Where do you see the biggest opportunities for innovation in downstream processing today?
It comes back to your initial question. Where's my biggest disagreement with the people common in the industry? And that is that there are many opportunities. Yes, we've been using chromatography for 100 years, but have we reached the limits of what can be done with it? No, not by a long shot. We have many opportunities to improve on the chromatography and the materials and the ligand that is responsible for collection of the moiety we're separating. We have many opportunities to reduce costs, to have higher efficiency, to have higher yields. It's a matter of changing materials, changing conditions, understanding the science behind it. We're very fortunate to work with some great chromatography experts in the world. I've been very fortunate. Because of my interest in chromatography, I've kept up these connections around the world and have personal friendships with Professor Shuichi Yamamoto in Japan and Alois Jungbauer in Vienna and many others who we work with closely to push the science further. What are nature's real limitations? What are the artificial limitations that people have done so far, but why can't we go farther?
Yeah, I like that. That's a very good approach, Scott. And I agree, there is still a lot of room for improvement. Think back of what's, for instance, going on in the biologic space with the continuous chromatography. That was a very big push into the right direction. Just out of curiosity, would your technology also work in a continuous mode?
Oh, yes. We have programs underway. I think it's great that especially for cell therapy, people have developed these systems where it's all self-contained. You start with the patient sample and you end up with the drug to give back to the patient. And yes, we want our chromatography as part of that for the purification step and used in that process. Continuous chromatography is very much of interest in the world today. We're giving a presentation at an international conference on integrated continuous bioprocessing coming up in a few months. So yes, one opportunity that is growing. Also, again, the new therapy modes that are growing. There are many opportunities in this industry.
I would like now to circle back to your story and your decades of experience, Scott. What are some key lessons you learned building companies and also operating in Asia? Many of our listeners are more located in Europe, North America. We do have a growing listenership in Asia, which is amazing. But what are some exciting things you have learned working extensively in Asia?
I have very much valued opportunities in Asia. I first went to Asia when I was an undergraduate. I went to Japan and lived there for 2 years as an undergraduate, learned the language, learned the customs of the people. It was a great experience. When you're young, when you're only a teenager and you live overseas, life teaches you some tremendous life lessons. And then I lived in India, I lived in China. I also worked in the US and Germany. Every country has its own business practice, and one must learn the business practice. I was living in India, working for Reliance Life Science. We built a nice plant there to make antibodies. It was very difficult. The business practice was very difficult. I thought, well, I'd like to stay in Asia. I could live in India or I could live in China. But I realized I couldn't do both. And I had all these friends inviting me back to China. So I said, well, I'll go to China. I'll see how it is. We were very successful. Innovate Biologics is now a $10 billion company. And that's not because of me. It's because, well, I hired the right guy to be the CEO. I brought my friend in. He was working for another company. I said, I want you to talk to my friends at Fidelity, talk to them about why we can really make this company successful with you as the CEO. And he did it, and he's still there. And smartest thing I ever did, hire him to be my boss. Asia is just a wonderful place, nice people everywhere, but every place is different. Again, I like the industry because it's nice people. I've been taken advantage of a few of times, but I'd rather be taken advantage of sometimes than to not be nice and kind to other people. Life's too short.
I love that. Life is too short. Experiment, try new things. I'm also curious, Scott, because you have worked in both startup companies, smaller companies, and in corporate biotech and pharmaceutical. What are some key leadership experiences you're now using as you're now building this new chapter at your current company in Singapore? And how are you using that and leveraging that?
Well, I'll always be grateful for the experiences that I learned in every company I worked at. And the experiences working at a large company like Abbott, where we had 10,000 employees at one site, were very different from the ones I learned at Chiron when it was only 230 people, or BioChromatographix, which is now only 8 people. But it's been great. At the big companies, you learn how an integrated operation works together. So marketing is in charge of manufacturing schedules, and engineering is in charge of building. And if you want money, you can get it only if you meet certain requirements and you've done your analysis properly. So we still do those analyses. We had one discussion this week. We're looking at alternate laboratory space. You have to respond to the analysis. If the analysis says, oh, your plan of action is not going to work and it's too expensive, you should do something else, you change, you do it. I gradually went from larger companies to smaller companies. The large companies, there was enough money to do anything we wanted to, but we didn't have any permission. The difference between spending $5,000 and $5 million was 3 signatures on top of the first 10. You had to get the first 10 no matter what you did. In a small company, you have all the authority you want and no money. So you have to do it all yourself. You have to wear many hats. You have to figure out how to do this. I remember working for a small company and we were getting ready to do clinical trials. I said, well, where are the case report forms? And somebody said, we don't have case report forms. I said, we can't start a clinical trial without case report forms. I was manufacturing. So I said, okay, look at case report forms. Oh, I worked with clinical. We got the case report forms done in time for the trial to start. Because in manufacturing, you know how to get things done. We get things done. That's what we do. Great experiences and some great people, just wonderful people. And another thing about this industry is you're really fortunate to work with the same people again and again as you each change companies. And you go back and say, you know, I really like working with you before. Let's work together again.
Yeah, biotech is a small world, even though we work globally.
In every country and internationally.
Scott, what advice would you give your, let's say, 40-year younger self to succeed well in biotech?
I'd worry less. I just worry less. There are always opportunities. You know, you always worry about, oh, is this the right choice? It's not a choice between right and wrong. It's a choice between right, and right. Okay, the hardest choices are between what's good and what's good. Thank you. Just do it.
I love that. Smart biotech scientists, just do it. Just do it. Don't overthink it.
Yeah, don't overanalyze. There are always choices, and there are always mistakes. We all make mistakes. I try not to worry about those either. If you hurt somebody, that's harder. I try to avoid hurting other people. I'd rather be hurt than to hurt other people.
Scott, before we wrap up, what burning question haven't I asked that you are eager to share with our biotech community?
Oh, I would just like to add that one of the hardest things with the new technology is educating customers as to why it's necessary and how they benefit from it. So we have an extensive outreach program, publications, presentations, website, white papers. We never talked about chromatography in terms of convective terms, even 10 or 15 years ago, because it wasn't important. Now it's very important, but it was important then. We just didn't recognize it was important. Education, helping people understand, that's part of the challenge. It's part of the challenge with any new technology. One of the challenges we have is understanding the physics behind what drives the limits of what we're doing. And if we don't understand that, it's very difficult to know what is the limit and how do we get past it. Is it new material? Is it new chemistry? Is it the thermodynamics? Is there something we can do to make the reaction more attractive? These are all good questions, and that's part of science, right? We ask questions, we look for answers, and if we don't get the right answer, we try again. Actually, in our laboratory, we have a shared laboratory space. There's a sign on the wall that says, experiment, fail, learn, repeat. And I think that's a good model.
Yeah, that's an excellent model. And it's so key to understand what you're doing. Process understanding or technological understanding is key, is fundamental. And I completely agree that you need to understand what are the technological limits of what you're developing.
I think there's the mindset to learning. And if you're afraid to fail, it's very hard to learn. If you have to be right all the time, well, Enron, their executives were like that. And the crash and burn, spectacular. We want to learn. That means we have to be willing to fail.
Yes, absolutely. We want to learn. This has been great, Scott. I very much enjoyed our conversation. With all that we've covered today, what is the most important takeaway you want our listeners to walk away with?
Well, I hope they enjoy their work. I enjoy many things besides the science. I enjoy music and reading and literature and painting and all of this art. But I enjoy my work because it's a challenge and I work with nice people. And I just hope everybody has an opportunity to enjoy their work. And I would worry less if I did it over again.
Scott, where can people connect with you?
So we have a website, biochromatographic.com, and we're happy to connect on my LinkedIn. Look up my name on LinkedIn. Scott Wheelwright. Wheelwright is spelled like it sounds. Wheel and Wright. It's a person who makes wheels. So there's a middle W. If you spell it right, then you'll find it. I'm happy to respond.
Fantastic. Smart biotech scientist. I will leave the link down in the show note. Go reach out to Scott, learn more about what BioChromatographix is doing. And Scott, thank you very much for being on the show today. It has been a huge pleasure talking with you about the advances in chromatography.
Thank you, David. I've enjoyed it very much.
These were fantastic insights from Scott Wheelwright on the future of chromatography and bioprocessing innovation. From practical evaluation criteria to entrepreneurial lessons learned across decades, we have covered game-changing strategies for downstream processing. Are you ready to revolutionize your bioprocess development? Schedule your free consultation at brühlmannconsulting.com to get answers to any process development challenges you may have. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.